From the campus of Stanford University.
People are worried about data. They're worried about their privacy and their security. They should be. We need secure systems.
This is the future of everything.
But we can't have a system that closes that data off. It is too rich of a source of inspiration, innovation, and discovery for new things in medicine.
With your host, Russ Altman.
Today on The Future of Everything, the future of brain cancer, therapy, and response. Few things create more fear and anxiety than the idea of brain cancer. I'm a physician, and I know that when a patient comes into my office complaining of a headache, what they are really number one worried about is, do I have brain cancer? And so it's very important to reassure them appropriately after appropriate tests that they don't have brain cancer, because this is one of the things people fear the most. The brain is a special organ. It has 100 billion cells. They're connected. They communicate with one another to create all of the amazing capabilities of human thought, emotion, and action. And it develops, right from the very start during gestation and early childhood through a complex series of growth, interactions, even pruning of cells that are not necessary, and refinement of the system. This allows cells to make decisions about where to go and how to make their connections, and the whole physical layout of the brain takes all of this time during gestation, childhood, all the way into young adulthood, and, well, hopefully that some things useful happen even in aging. It's carefully and it's beautifully orchestrated, but it can go wrong. And when it goes wrong, it can lead to brain cancer. And, of course, one of the more tragic parts of brain cancer is when it affects children. Now, we have a variety of treatments, and we might learn a little bit about those today, but these almost always have toxicities and side effects that limit their utility and their effectiveness, and also have implications in the long term for the patient. Some of these side effects may affect the core functions of the brain, including even cognitive impairment after treatment. And brain cells cannot typically grow back. People are working on that, but currently, if cells are lost, they don't come back, and so it's important to understand and prevent any kind of neurological damage during treatments. Fortunately, the modern tools of research are shedding light on these issues and giving us causes for hope. Dr. Michelle Monje is a professor of neurology, neurosurgery, pediatrics, pathology, and psychiatry at Stanford University and studies brain cancer, particularly in children. She searches for new therapies, and she also studies the effects of those therapies and uses all of that to understand fundamentals about how the brain develops. Michelle, brain tumors in children are particularly tragic and challenging. You know that better than I. What progress has been made, and what are the most promising developments?
So that was an excellent introduction, Russ.
Thank you.
Brain cancer in children is one of the worst diseases that we can imagine. When I saw this disease first in medical school, I just couldn't turn away from it. And so I've dedicated my career to trying to take better care of children with brain tumors. And I can say that over the last decade, we've made, as a field, just enormous progress. When I started studying these tumors 10 to 15 years ago, we knew very little about, the mutational profile that is driving the initiation of the cancers, about what these tumors need to grow, about why they exhibit such a remarkable but poorly understood spatial, meaning different parts of the brain and central nervous system, and temporal, meaning at certain ages, kind of pattern. So certain cancers happen in certain places in the nervous system at certain times of childhood and adolescent development. But what has been governing those patterns has been really poorly understood.
Is the growth of the cancer related to the development of the brain? Are those going part and parcel, and is something going wrong, basically?
So that's really the hypothesis that I started my research program with, that if we better understood neural development and neuroplasticity during childhood, adolescence, young adulthood, and on into, old age, that we would understand more about how these tumors emerge and what we could do to effectively treat them. Because what has been so frustrating is that, for the worst acting brain cancers, we have very limited therapeutic options at present. That is rapidly changing, which is so exciting and so wonderful to see. And for the tumors that we can effectively treat, we use such big guns, if you will, that there is often, if not usually, lasting damage to the nervous system that really affects the individual for the rest of their lives.
They often say that you're trying to kill the cancer before you kill the patient, and it must be especially worrisome when the toxins that you're delivering, and when I say toxins, I mean the very powerful chemotherapies, when those are going into the brain, which is such a tender and critical organ, it just makes you worried.
It makes you worried, and for good reason. Our therapies are presently, they're the best we have. We need them. They're life-saving. I'm not at all advocating not using the therapies that we have, but they are devastating, and they induce long-lasting damage that is also, or has been, really poorly understood. And so, the overarching goal in the field is to develop more effective therapies for all kinds of brain and spinal cord cancers, and for those therapies to be safer, more specifically effective, and or to develop ways to regenerate the nervous system and repair the damage that these therapies are doing.
Yeah, so with all your knowledge, and you're both a clinician and a researcher, how do you decide where do you focus your efforts, and what do you find to be the most promising directions? Because obviously that's what you spend all your time thinking about, so you can use your efforts productively.
Right. So some of the most exciting developments in oncology in general and in childhood brain cancer specifically have come from number one, a deeper understanding of what is really driving these cancers, and that comes back to fundamental neural development in understanding how we can perhaps interrupt some of the factors that are enabling and encouraging the growth of the cancers. And again, that comes back to understanding neural development and plasticity. And the other really enormously promising area is in immunotherapy and leveraging the power of the immune system to try to specifically eradicate the tumor cells. So
the immune system is in the brain. It's not walled off in some way.
That's absolutely right. And actually, discoveries in basic neuroscience and basic immunology have elucidated that, in fact, not only the resident immune cells of the brain, which are called microglia, they're a resident tissue macrophage cell, we've known about their presence for some time, but in addition, there are lymphocytes that are, we used to think about the brain as kind of a immunoprotected site.
Yes.
But in fact, lymphocytes are traveling to the brain, they're regularly surveying the brain, they might be contributing to normal processes of neurophysiology, and those lymphocytes are in fact being drained from the brain through a lymphatic system that was only discovered in the last few years. So we didn't even know that there were lymphatics in the brain, but it turns out there is a neurolymphatic system.
Yeah, so we've heard in the popular press about these amazing immunotherapies where you can get your own immune system, the patient's immune system, to realize that the cancer is bad and to specifically target it to kill preferentially these cells. And it's not easy because these cells also have hallmarks of being part of the patient.
That's right.
And your system, your immune system is trained, don't attack yourself.
That's right.
So this has now started to play out for childhood cancers.
So this has the immunotherapy for some other non-central nervous system and
melanoma is a big one.
Melanoma is a great example. Leukemias, lymphomas. There have been enormous strides forward in terms of effectively leveraging the person's own immune system to recognize the cancer. That has not yet played out for brain tumors, adult or pediatric, although there are some strides forward. And what we're beginning to understand is that some brain cancers allow the immune system to see it, and others really hide from the immune system. I think of pediatric brain cancers as being somewhat ninja-like. They are hiding from the immune system. They're diffusely, quietly infiltrating much of the central nervous system. And so part of our challenge is to alert the immune system to this invader, if you will, or to deliver an immunotherapeutic product like an adoptive cell, an immune cell that we've taken out, altered, and put back so that it can see the cancer and specifically eradicate it.
Kind of give the immune system a little bit of help in saying, hey guys, these cancer cells may look like they're normal neural cells, but they're not. They're the bad guys.
Right.
Go after them.
So the trick is to find the signal that allows the immune system to see the cancer cells as different from the rest of the brain, because as you said, they're very, very similar. And so understanding what these cancer cells are expressing on their their surface that can be seen by the immune system, whether it's the person's own immune system or an immune cell that you've given some help to, is a challenge. And there's been, at least in the laboratory, some huge strides forward in that for childhood brain cancer. And I hope to see some of those things come to clinic very, very soon.
This is The Future of Everything. I'm Russ Altman. I'm speaking with Dr. Michelle Monje about brain cancer, especially in kids, and the outlook. So until these immune therapies really develop, we're kind of stuck with these, what I would call toxic therapies, where you're basically trying to kill the rapidly dividing cancer cells. But you've taken an interest not just in getting those therapies to work, but also to understand their long-lasting effects. These are children, and they're going to have hopefully 50, 60, 70 years of life afterwards. And you've begun to ask the question, what is it doing to the brain over the long haul? So what do we know about that?
Right. So, what we have, I think, intuitively understood by taking care of children who've gotten these therapies is that some process of neuroplasticity or neurodevelopment is fundamentally altered after these therapies. But why that happens has been very unclear. And so my laboratory has been studying this for a number of years. And a central theme that is emerging is that many of our cancer therapies change the activation state, if you will, of the resident immune system in the brain, the microglia, these tissue macrophages. Now, in the healthy brain, microglia contribute to a number of really important processes, like you mentioned, connections in brain cells are forming during childhood, and then they get pruned back. Microglia prune those connections, which are called synapses. Microglia contribute to development of the myelinated infrastructure of the brain, to the white matter. They likely do additional, trophic growth-promoting things that we haven't understood and are working to understand.
I'm struck that I shouldn't think of the brain as just sitting there. It's a very active organ.
Absolutely.
With lots of things going on, even though it looks like it's just a blob of jelly.
Right. It's constantly changing, especially during childhood, but well into adulthood. And if I could just take a side, make a side point about that, the brain is not only actively developing, but how we use it changes the way it develops. And we kind of know that because when we practice something, when we study, we change our nervous system's ability to perform a task or to remember, a fact or to, develop a skill.
Yeah,
and
so something must be changing because I couldn't do it last year and now I can do it this year.
That's right. And so part of what's changing are the details of those connections between brain cells, and other things are changing too. And so part of our research program has been focused on understanding how activity in the nervous system, which is induced by experience and by practice, is influencing not just those connections between cells, but also other fundamental things about the structure of the brain, and specifically the insulation around the axons that contributes to white matter. The
axons are the appendages of the cell that help the cells communicate.
That's right. So the sort of electrically active, cells in the brain, which are neurons, connect to each other through these long processes called axons, and then they connect at the synapse. And in order for a signal to travel effectively between one neuron and another, or one part of the brain and another, that signal has to go along an axon. And that process is greatly facilitated by the establishment of these the myelinating infrastructure of the brain. So myelin is formed by cells.
It's almost like an insulator on a wire to keep
it's very much like that.
Keep the signal moving.
It's exactly like that. Actually, it decreases transverse, capacitance and increases the speed of the signal transduction along the wire, or the axon, if you will. And that myelin covering on the axon is formed by a cell called an oligodendrocyte that's broadly in the class of cells we call glial cells. And
glial, we all recognize that we all hear about glioblastoma and glioma.
That's right.
So I'm thinking that this is a cell that can cause us trouble.
That's right. So this is a cell that can, on the one hand, contribute in really important ways to development and plasticity. And that's one thing that we have focused a lot on, and understanding how activity regulates the changes in those cells both during childhood development and well into adulthood. And the one hand, that's a cell type that can give rise to these glial cancers like glioma. And on the other hand, it's a type of cell that is very susceptible to damage by chemotherapeutic agents and other cancer therapies.
This is the future of everything. I'm Russ Altman. I'm speaking with Dr. Michelle Monje, who's telling us about these glial cells that are both our best friends and are also can be our enemies. So what have you learned about how these cells respond to chemotherapy? And I guess they're very sensitive.
They're very sensitive, but they're sensitive in a very interesting and perhaps therapeutically targetable way.
Oh,
and
so this is good news.
So this is good news. And so I mentioned microglia, which are one major class of glia. I mentioned oligodendrocytes and their precursor cells. That's another major class of glia. There's also a third class of glia called astrocytes. And it turns out that some commonly used chemotherapy medicines as well as radiation induce activation of microglia so that they are no longer trophic. Instead, they are inflammatory.
So what does trophic mean?
Growth promoting.
Growth promoting.
Growth
Okay.
and plasticity. They're not doing the good stuff. Now they're in a defense mode.
Okay.
And when they're in that state, there's some unintended consequences. And so work from Ben Barris's lab here at Stanford a few years ago demonstrated that when microglia are activated, they can activate this third class of glia called astrocytes to become toxic rather than supportive.
Okay, sounds bad.
It's bad. And as well.
So the chemotherapy is activating the glial cells that are then turning on these toxic cells.
Right. So they're changing the microglia and the astrocytes after at least the kinds of chemotherapy medicines that we studied, perhaps not all chemotherapies, but we studied one in particular called methotrexate, they become activated, they stop being helpful, they start to be somewhat harmful, and they disrupt very dramatically the function of these myelin-forming cells. What is exciting is that if we can normalize the microglia, and the way that we've done this experimentally is to actually remove them for a period of time, which we can do with medicines that target them.
Right.
That helps the astrocytes return to their normal state and become supportive and not toxic, and it normalizes the function of these myelin-forming cells in a way that rescues cognition, at least in mouse models.
No, so I see you're painting a picture now of therapies that are treating the cancer but have these unfortunate side effects, but there may be this secondary therapy that can try to mitigate the side effects. So you're still getting the good effects of the toxic
That's right.
medication, but then you're adding other either medications or maneuvers of some kind to try to minimize their long-term impact on the brain's
That's exactly
processing.
right. That's exactly right. So as we start to understand what's going wrong after cancer therapies, we can start to intervene therapeutically. And right now we're working in the lab to understand what our time frame is. Is this something we have to do right away, or can we come and try to help people who have been suffering from the cognitive effects of cancer therapy 10 years later and try to improve their cognition through rescuing some of these processes of development.
And I'm struck that by studying the cancer treatments, and then the mitigation of the cancer toxicities, you're probably learning very fundamental things about how the brain develops that may have implications for things far beyond cancer.
That's right, which is one of the wonderful things about basic science is you study one problem and you start to see connections to other problems. And so, this dysfunction of glial cells is something that is now being recognized in a number of different neurological diseases. And so microglial activation, which we found after radiation, after methotrexate chemotherapy, also happens in most, if not all, neurodegenerative diseases like Alzheimer's disease. And so as we study one problem, we're really learning things that are applicable to many.
Well, we'll definitely have to get more into that. This is The Future of Everything. I'm Russ Altman. More with Dr. Michelle Monje about brain cancer, brain cells, brain toxicity, and what we can learn about diseases that may have nothing to do with cancer next on SiriusXM Insight 121.
From the campus of Stanford University.
People are worried about data. They're worried about their privacy and their security. They should be. We need secure systems.
This is the future of everything.
But we can't have a system that closes that data off. It is too rich of a source of inspiration, innovation, and discovery for new things in medicine.
With your host, Russ Altman.
Welcome back to The Future of Everything. I'm Russ Altman. I'm speaking with Dr. Michelle Monje about neurology and what we can learn from neurology about brain cancer and from brain cancer about neurology. So one of the things that your lab does is study the very basics of how neurons function, even in the normal brain. How do those studies inform your understanding of how cancers grow? Because obviously the thing that's worse about the cancer is that they grow and they grow out of control and they start taking up space, they crush other parts. So the growth of the cancer really is critical. And I guess it's related to the basic processes of how normal brain tissues grow.
Absolutely, absolutely. And so that's actually that idea is really one of the guiding principles in what we do. And so because my laboratory and my clinical practice focus on glial malignancies, gliomas like glioblastoma, which also happens in children, although there's some differences between the adult and the pediatric disease, we wanted to understand at a very fundamental level what promotes the replication and growth of normal glial cells. And thinking about, the origins developmentally of these glial malignancies, what promotes the proliferation and growth of specifically the glial cells that are perhaps giving rise to these cancers. And so one of the, I think, really exciting things about the way that the brain, functions and changes and is plastic and adaptable is the discovery that experience through neuronal activity influences the proliferation of these glial precursor cells. And this actually is fundamentally important to the way that experience and activity changes brain structure and therefore function. And we call these changes in the glial infrastructure myelin plasticity. Well, it turns out that the cancers hijack those very mechanisms that are normally playing out to promote adaptability and plasticity of the brain. And so that
seems quite diabolical.
It is quite
cells
diabolical.
That now respond to my learning and my interaction with the environment, the cancer cells have figured out how to imitate some of that to get the glial cells to do things that they shouldn't be doing.
That's right. And so when you think about these cancers, we often think about cancer as a ball, as a nodule, right, growing. But most of these glial malignancies, particularly in childhood, but also in adulthood, really diffusely invade the brain. And they are intimately interacting with all of the other cells that are present in the brain that are normally contributing to these processes of growth and plasticity. And so as we have dug in and drilled down to understand the mechanisms by which brain activity can promote brain cancer growth, we've uncovered some really exciting therapeutic targets, things that we can target to disrupt this ability of the cancer to hijack these growth signals that are produced as a result of activity. And one of these targets is resulting in a clinical trial that we're hoping to open in late 2019. So I'm really excited about this new direction of targeting these microenvironmental dependencies, if you will, the dependency of the cancer on the brain environment in which it's growing.
And it sounds like these won't be the kind of blunt instrument, toxic, kill the cancer before you kill the patient. These will, I don't know if the word precision, but it seems like this is a much more targeted kind of way to treat. So can you give us a little feel for what you're, how do you trick these cells into not being bad?
Yeah, so,
or as bad?
So one of the signals is a molecule, a protein that gets released from normal brain cells as a result of brain activity. So whenever the brain is active, whenever neurons fire, this molecule gets released. And it gets released into the environment where the tumor can use it to grow through the activity of an enzyme that actually clips it off of the cell surface and lets it be free to bind to the cancer cells. So we can prevent that, the molecular scissors, if you will, the enzyme that clips that molecule so that we can prevent it being available for the cancer cell.
And so the cancer cells never get the signal to grow.
Right. That signal to grow. And there are many signals to grow in the brain, but this one seems to be particularly important. Now, that's not to say that this particular therapy will be without any neurological side effects, because we know that the clipping off of this molecule is part of normal brain physiology, but we think that it'll be a much more limited and specific, side effect profile.
This is The Future of Everything. I'm Russ Altman. I'm speaking with Dr. Michelle Monje about new approaches to take advantage of our understanding of neurology and neuroscience to more precisely confound the ability of cancer to grow. In the end of our discussion, I just wanted to go to the clinical side. So you're doing this very detailed, very profound work in neuroscience, but you're doing it for kids, and you see these kids. So What is the experience of a child like with these terrible diseases? What have you seen in terms of the experience that they have acutely with the disease and then the sequelae afterwards? And how are kids responding to these cancers?
So I have to say that my patients are enormously inspiring, and everything that we're doing is really to try to help them. Kids are remarkably resilient. You know, they're brave. They fight these cancers. They come to terms with their outcome when the outcome is not going to be good in a remarkably brave and mature way, typically towards the end. They start to think about how their experience can help other children. And they think a lot about their family. And, you know, there's a very well-known case that I think really illustrates this principle of a little girl who lived in the Midwest. She was not a patient of mine, but I know her parents very, very well, as they've really engaged in trying to support the research community for this disease. And she had one of the universally lethal forms of brain cancer. And knowing that she was going to die, she left Post-it notes throughout her house, hidden for her parents to find.
Oh my God!
And it's been about
How old
12.
was she?
She was 6.
Oh.
Yeah.
And they're not still finding them? Or?
They are still finding them. And there's actually a wonderful book that this amazing couple wrote called Notes Left Behind.
How do you explain to a child what's going on in their brain when you have to break the news to them and to their parents? And what are the kinds of questions they have?
Yeah. So number one, it's really important that kids have a sense of what is happening. It's they have to know that they have cancer. They have to know that it's serious. We often want to protect children from scary things, right? Tell them there are no monsters. But you know, it's really important that children understand, at least at the level that is appropriate for them, what is happening. And so it depends on the age, but we talk about spots, we talk about bumps, we talk about, you know, trying to fight the bumps, and we talk about the fact that we don't always have the right medicine to effectively fight that spot or that bump, but that we're going to take care of them throughout the whole journey. And they understand that. I think as time goes on and with conversations with their family, they understand it more and more. They often have really insightful questions. And particularly older children, you know, really focus on making sure that everyone around them is okay, making sure their parents will be okay.
Altruism in the face of this terrible disease. How do you, now I'm going to turn to the parents, how do you start to explain the incredibly complex underlying biology, not to the children, but to parents who are dealing with something terrible? This is tough stuff to understand. It's your work is the result of many years of study. How can you simplify? I don't even want to use that word almost, but how can you give them the rough idea of what's going on, especially when you're introducing them to new medications that are working in entirely new ways and you have to get consent from them to participate? Yeah.
So, you know, I think really doing our best to explain in English and not in med speak, you know, how these cells interact with each other and what we're trying to do to you know, make them behave better, if you will, is critically important. I recently created kind of in an effort to effectively, sorry about that, I lost my headphones, to effectively communicate the science behind, you know, chemotherapy-induced cognitive impairment, a short video, a cartoon, describing, you know, how cells that normally need to work together to effectively do their job, you know, might become dysfunctional when one member of that group starts to overreact to something.
This is what we've been talking about for the last 20 minutes. Right. So if somebody wants to Google that.
Yeah, so it's on YouTube. It was a part of what's called a video abstract on the Cell website. It's actually also on my laboratory website. So
Great.
if you
So
go
if
there,
they Google Michelle, Monje, that should get them.
That should get them there. Yeah.
Well, thank you for listening to The Future of Everything. I'm Russ Altman. If you missed any of this episode, listen anytime on demand with the SiriusXM app.